Second year of MBBS is supposed to be a very fun and easy year, but there is one subject which ruins all the fun. So hey everyone, welcome to this video. My name is Shoham. I'm a third year MBBS student at CNMCH Kolkata. In my second professional examination, I scored a distinction in the subject of microbiology while also doing this, and this. So I'm going to show you how to do exactly this without wanting to bang your head on the wall. Because trust me, a lot of us did want to do this in the beginning of the year. So this is going to be a straight to the point video of how I handle things personally and what I did personally to make the subject easier for myself. It's not going to be any vague advice like study every day or make notes or teach it to a friend. Because let's be honest, you probably knew all that and you did not click on this video for me to reaffirm what you already know. See, in MBBS, we're not really interested in the microbe itself because we don't care about the microbe. A lot of people waste their time studying about the microbe, everything about their layers, their reproduction, their motility. That's important, but we don't need to delve too much into that. What we are actually interested in that how it gets inside a human being, how it causes a disease in the human being, and how we can find out that that specific microbe is infecting the patient we have. So don't waste too much time studying about the microbe. Study what effect it has on the human being. Second is the resources. Luckily for this subject, you only need one book, and we have been provided this book. You probably already can guess what the book is going to be. It's Apurva Shastri. It is considered the Bible of microbiology by most second year students, including myself, because if you read this book, you don't need any video lectures. I personally haven't watched a single video on microbiology. Everything I've known, everything I've learned is from this one single book. And it gets even better. Everything in this book, everything you need for your exams, can be further condensed into one single notebook. This is the only notebook I've had for this subject. See, it's written Micro 1 because I thought it would be longer. And to my pleasant surprise, there has never been a Micro 2. Number 3. What to do when you first open a chapter? The basic idea is you first have to understand the layout of the chapter you are about to study. If you start reading the first line itself after opening a chapter, right at that moment, you have no idea what this chapter is about. You have no idea from what topic to what topic the chapter is going to jump. You have no idea what to expect. So you are basically going in blind. And when you do that, you are just reading a paragraph. You're trying to make it make sense. And when you start reading the next paragraph, you forget everything you've read. To avoid that, you first glance at the entire chapter. You skim every single page and read the broad headlines. Let's say you've opened this chapter, General Mycology. If you start reading from line 1, it's going to be very exhausting. Yes, these points are important, but you don't start reading from there right away. First, you look at the broad headlines. Classification of fungi, the ones in blue. Classification of fungi. Laboratory diagnosis. Then there is no other blue line, so you skip this part. Then you go to overview of infections. So basically, what you look at in your first reading is what are the major topics which are being handled by this chapter. After you've read the book, you take out your notebook and you make a flowchart like this. Major headings, and then the subheadings, and then whatever else is there. Now, by doing this, you have created a sort of skeleton of the chapter inside your brain. Now, if you start reading the chapter from line number 1, it will all make sense because now your brain has something to relate to. Okay, I'm studying this part of the chapter. I'm studying that part of the chapter. Your brain will be able to retain it much more efficiently than if you had just started reading from line 1 and going blind. Next is active recall. Everybody tells you about active recall, but nobody really tells you how to actually do it. Active recall is a method where you ask yourself questions and you test if you can actually answer those questions. This helps in training your brain to retrieve the information you have stored in it rather than just passively mugging up the textbook. Let's see how it works in action. This is an example of questions I've written for CNS microbiology. Now, what you can notice here is I'm not just asking what questions because what questions are just recalling facts. You have to ask yourself questions which will actually help you with the understanding. Your notes need to be as minimal as possible and cram in the most information in the most efficient way. Now, if you didn't understand what I just said, your notes need to be concise enough to fit in one notebook. In enteric fever, I've written it like this. I have written the symptoms: rose spots, nausea, myalgia, arthralgia, diarrhea. Then the unique features: nosebleed, hepatosplenomegaly, bradycardia, and coated tongue. Now, after you've done this, you've seen the symptoms, you will start thinking like a doctor. Okay, a patient has walked in my clinic like this. I've seen these symptoms. Now I'm interested to know what has actually happened to them. If you see the symptoms, you will actually be interested in learning more about the disease. And then it will help you to not get bored of the chapter. Number four, most important point, let's address the elephant of the room, which is lab diagnosis. This is what gives the most nightmares to a student, including myself. That was before I actually figured out what it was actually about. Lab diagnosis is finding out what microbe is causing a disease in a patient. What you have to understand is the microbe is present inside the human body. You have to take something from the human body, any part which contains the microbe, you have to take it to a lab and then you have to carry out a series of tests. Now, the tests are going to be, of course, different for different types of microbes, but one basic layout you can put in your head is number one, taking a sample. You have to take a sample of the microbe from somewhere in the patient's body. Now, from where? You have to know what system. If it's a bloodstream infection like typhoid, you have to take a blood sample. If it's a CNS infection, you have to take the cerebrospinal fluid. So, if you know what system a microbe is from, you can easily know what sample to take. Next is carrying the microbe from the patient's home into the laboratory. If somebody is coming to your house and taking a sample from your house, they have to carry that into the lab. And that process is called transportation. And then after it reaches the lab, you have to place it under the microscope. This is the basic layout of how lab diagnosis work. And after you've seen the microbe under the microscope, you will have a rough idea of what the microbe is. Because you cannot identify a microbe right from the microscope. You have to carry out some further tests. So the basic layout is sample collection, transportation, and direct smear. Direct smear means you're taking the sample directly and making a slide which you will view under the microscope and try to see what the organism is. After that, you know what kind of organism it is, so you know what series of tests you have to perform to actually confirm it. To make your life easier, there is a whole chapter on lab diagnosis in your general microbiology. So please read that. General microbiology itself is very important. So please don't skip out the first few chapters because you think there is no questions coming from there. But that is what will help you shape your understanding of the entire subject. The chapter on lab diagnosis is just what I think 18, 19 pages. So up until this, it was about how to read and understand the subject. Now, here are a few exam specific tips which will help you retrieve the information and produce them during your exams. A huge problem we face during our exams is we freeze up when we see the questions because it feels like we kind of know the content of the answer but we don't know how to begin it. So, what I have done, and it has worked very well for me, is I write the first few lines of every single topic in my notebook. I know what topics are there in my book. So if I assume that a topic is going to be asked in the exams, I take out my notebook and I write the first 2 3 lines of the answer as if it were a real question. So that if the question actually comes in the exam, we have already trained our brain to retrieve the information immediately. So MBBS students do have this quality that they can find one word or one line and they can write an entire page about it. So this will actually help you do exactly that. This is an example of how I wrote the beginning of the answer as if it were a real question. It's rotavirus diarrhea, as you can see here. This is another example of the morphology of Taenia solium and saginata. This question usually does not come in the exams, but this short introduction also serves as a very stupidly simple explanation of what this chapter is about. So if we haven't revised this and we are coming back after a very long time, this might help us jog our memory. Very useful tip which I can give you to help you recall your topics better is that when you're studying something new, you're and you come across a new term, try to describe it as stupidly simply as possible. Because when you're trying to recall something, the first question which comes to your mind is what that thing is. Let's say, for example, you're studying bacterial capsules. The first question which will come to your mind is what are bacterial capsules? Now, if you can't immediately answer it in your head, you will start to feel that you don't know the topic and you will start to lose confidence. So, to prevent yourself from losing confidence, every single thing you read, you have to answer the question what that thing is. Whatever topic it may be, you ask yourself, if I ask myself later what that topic is, would I be able to answer it? Let's take an example. I gave you the example of capsule and slime layer of bacteria. So this is the stupidly simple definition I wrote of this in my notebook. The reason behind this is you might know plenty of things about bacterial capsules, but if you don't know the basic definition, if you don't know line number 1, even if you know 10 other things about bacterial capsule, you will not be able to begin your answer. So you must know the beginning of the answer to actually access the rest of the answer from your head. And last but not least, the same old advice, study every day. Because even if you study one page, it's still useful. If you lose the habit of studying, you will not be able to catch it back. Like what's happening with me right now in third year. So I hope you found this video useful. It will definitely help in your microbiology preparation. Like and subscribe. All the best.